Welcome to the Deep Dive, the show engineered to give you a true shortcut to being well-informed fast. Today, we're not just scratching the surface, we're taking a full-fledged deep dive into the foundational functional organization of the cell. Think of this as your essential guide to understanding the molecular building blocks that allow all complex biological systems to operate.
That's right. When we talk about physiology, it's easy to picture entire organs or, body systems at work. But to truly grasp how your body functions, you really need to understand the intricate homeostatic mechanisms happening at every level, right down to the molecules inside your cells. For this deep dive, we're drawing key insights from Boron and Boulpaep's Medical Physiology because it lays out the cellular machinery that allows us to persist despite constant environmental changes.
Yeah, our mission is to transform these detailed, sometimes dense concepts into clear, engaging, and academically accurate explanations. We want to make complex physiology accessible, connected to real-world clinical relevance, and build concepts from the ground up, step by step. And since this is an audio deep dive, we'll paint all the mental pictures you need, no visual aids required. So let's begin with the cell's essential outer shield, the plasma membrane. Picture any living cell, whether it's a tiny single-celled organism or one of the countless neurons packed in your brain. Each one's survival hinges on maintaining a unique internal environment, profoundly different from its surroundings.
Absolutely. The cytoplasm, the cell's inner world, is this complex solution brimming with proteins, nucleic acids, ions, sugars. This precise composition is maintained at significant metabolic cost. Without a barrier, this carefully regulated balance would just be lost to diffusion in mere seconds. This is where the plasma membrane comes in. It's the cell's outer skin, acting as a critical selective barrier. It's impermeable to large molecules, ensuring they stay put, but carefully controls the passage of smaller ones like ions and nutrients.
So it's not just a passive fence, is it? It actively participates in shaping the cell's environment.
Far from it. A core insight here is that the membrane isn't just a filter, it's a dynamic gatekeeper. It actively accumulates nutrients, often against their concentration gradient, using processes like active transport. This requires metabolic energy, and it's this ability to create and control gradients that powers, basically everything.
Right, like neuronal signaling.
Exactly. From your neurons generating electrical signals to your kidneys filtering waste and reabsorbing vital substances. Without this precise gradient control, life as we know it simply wouldn't be possible.
That's a profound point. So how is this sophisticated skin actually constructed? Much of our understanding comes from early studies on red blood cells or erythrocytes. These cells are essentially just a membrane wrapped around hemoglobin-rich cytoplasm, making them ideal for analysis. Scientists found the membrane is primarily made of lipids and proteins, with phospholipids being the most common lipid.
Phospholipids are the fundamental building blocks here. They have a glycerol backbone, 2 fatty acid chains, a phosphate group, and a head group, which gives each phospholipid its unique identity. Think of them as having a dual personality. They're amphipathic, they have a hydrophobic tail, these long nonpolar carbon chains that, hate water, and a hydrophilic head, which are charged, or polar groups that love water.
And this dual nature leads to something remarkable when they're in water, right?
Indeed. These amphipathic phospholipids spontaneously self-assemble. At higher concentrations, they form a bilayer structure, 2 parallel sheets with their hydrophobic tails facing each other in the middle, creating this sort of hydrophobic sandwich. The hydrophilic heads face the water on both the inner and outer surfaces, forming an incredibly stable barrier.
So it's this perfectly organized sandwich, but like you said, it's not rigid. The phospholipid bilayer is actually a remarkably fluid structure. Individual phospholipid molecules can easily diffuse laterally within their own layer, just sliding past each other.
Exactly. This fluidity is influenced by what we call transition temperature, the point at which the membrane shifts from a fluid sol state to a more rigid gel state. Longer saturated fatty acid chains pack tightly, requiring more energy to move, thus raising the transition temperature. But kinks from double bonds or shorter chains disrupt this tight packing, which, it lowers the temperature required for fluidity.
And what about cholesterol, the famous molecule? How does it fit into this delicate balance of fluidity?
Cholesterol. It's another key lipid in animal cell membranes. At moderate concentrations, its rigid steroid ring actually makes the membrane less fluid by partially immobilizing the fatty acid chains. However, and this is interesting at high concentrations, cholesterol can surprisingly increase fluidity by disrupting phospholipid packing. Bilayers with mixed lipids can even form distinct lakes of fluid and rigid regions.
Here's a fascinating insight. While phospholipids easily move sideways within their layer, moving from one side of the bilayer to the other, a flip-flop, is extremely rare. It would take enormous energy to drag their water-loving head through the oily core. But cholesterol, with its small hydroxyl head, can actually flip-flop relatively rapidly.
And this brings us to permeability. Pure phospholipid bilayers are highly impermeable to charged molecules like ions, Na+, AAK+, Cl, Cl2+, and large water-soluble molecules like proteins and sugars. This is fundamental for maintaining the cell's distinct internal environment. Yet small uncharged polar molecules like oxygen, carbon dioxide, and even water itself can cross fairly freely. It's quite surprising.
That's amazing. How did we even prove it was a bilayer in the first place? Back in 1925, Gorder and Grendel extracted lipids from red blood cells. They spread these lipids on a water surface and found the monolayer they formed covered exactly twice the surface area of the original red blood cells. This strongly implied a bilayer. Later, electron micrographs visually confirmed this, showing two dark lines where stains bound to the hydrophilic heads separated by a light core.
And a critical detail here is that the two sides or leaflets of the plasma membrane aren't identical. There's a distinct lipid asymmetry. The inner leaflet, facing the cytoplasm, has different phospholipids like phosphatidylethanolamine and phosphatidylserine than the outer leaflet, which has phosphatidylcholine. This asymmetry is established during biosynthesis and is crucial for protein interactions and internal cell signaling pathways.
Okay, so if phospholipids are the basic structure, then membrane workhorses are definitely the proteins. These are the membrane's key functional components, broadly classified as peripheral and integral. Peripheral proteins are loosely attached, easily removed by just washing with salt, but integral proteins are tightly embedded, often spanning the entire membrane multiple times, and you need detergents to get them out. Some are even lipid-anchored, covalently attached to a lipid that inserts into the membrane.
Yeah, integral proteins, especially the transmembrane ones, pass through the membrane's hydrophobic core by using segments that are typically hydrophobic alpha helices. These helices are perfectly suited to the oily environment, with their side chains interacting with the lipid tails. And these proteins can have really complex topologies, meaning their ends, the N or C termini, are specifically oriented toward either the cytoplasm or the extracellular space.
And like the lipids, these proteins are also mobile, right? I remember reading about this experiment. Fry and Adedin in 1970. They fused human and mouse cells, labeling their surface proteins with different fluorescent dyes. Initially, the labels were separate, making the fused cell look half and half. But over about 30 minutes, the dyes intermixed, showing that membrane proteins diffuse and intermingle, similar to phospholipids, though maybe a bit more slowly.
That's a classic experiment, and a key point here is that unlike lipids, membrane proteins almost never flip-flop from one side to the other. Their fixed topology is essential for their function. Their movement can also be restricted by attachments to the internal cytoskeleton, we'll get to that, or actively driven by molecular motors.
So what exactly do these essential membrane proteins do? Well, a big role is as receptors. Integral proteins are perfectly positioned to transmit signals across the membrane. Think of a hormone binding to a receptor on the outside of your cell. That binding triggers a shape change in the receptor, which then relays a signal to the cell's interior, often via second messengers. This is how your cells communicate and respond, to their environment.
Absolutely. They also function as adhesion molecules. These are vital for physically connecting cells to their surroundings. Cell matrix adhesion molecules, like integrins, link cells to the extracellular matrix, influencing cell shape and growth. Then there are cell-cell adhesion molecules, such as cadherins, linking cells to each other, critical for forming stable tissues. Clinically, the loss of these adhesion molecules is actually a hallmark of metastatic tumor cells, allowing them to break away and spread.
Right, that makes sense. And remember how we noted pure lipid bilayers can't transport substances uphill against their gradient? Integral proteins provide this capability through transporters. These can be channels, carriers, or pumps. They basically create hydrophilic pathways through that hydrophobic membrane. Pumps, for example, use ATP energy to actively move substances against their concentration gradients. It's an energy-intensive but vital process for maintaining cellular balance.
And the ingenuity behind these hydrophilic channels and hydrophobic proteins is, well, it's remarkable. The membrane-spanning alpha helices that form them are often amphipathic. That means they have both hydrophobic and hydrophilic surfaces. They pack together so their hydrophobic surfaces face the surrounding lipid, while their hydrophilic surfaces line a central pore, forming a selective pathway. A potassium channel, for instance, is formed by multiple subunits, contributing these amphipathic helices, creating a very precise selective pore just for potassium ions.
Many integral proteins are also enzymes. Ion pumps, for example, are enzymes. They hydrolyze ATP to power transport. But numerous other membrane-bound enzymes exist, particularly in the intestine. They perform the final stages of digestion right there at the cell surface, conveniently close to transporters. It makes the membrane an incredibly efficient two-dimensional reaction center.
And connecting this to the bigger picture, some lipid-anchored integral proteins on the inner leaflet are crucial for internal signaling and growth regulation. For instance, some oncogene products, proteins involved in cancer, actually need this lipid modification to induce tumors. This insight is a major area of medical research, as inhibiting this attachment could potentially prevent tumor formation.
That's fascinating. Finally, we have peripheral proteins that form a subcortical cytoskeleton just beneath the plasma membrane. This meshwork of proteins, like spectrin and actin in red blood cells, provides structural support and resilience. In red blood cells, this network is absolutely critical for maintaining their biconcave disc shape and preventing fragility. Mutations in these proteins can lead to fragile, easily torn red blood cells.
And this isn't unique to red blood cells. Similar submembranous meshworks exist in neurons and epithelial cells, playing a critical role in organizing the plasma membrane into distinct functional domains, kind of like specialized neighborhoods on the cell surface.
Okay, now let's step through that sophisticated membrane and zoom inside the cell. It's not just a simple bag of fluid, but a meticulously organized microcity filled with specialized membrane-enclosed organelles, each with its own crucial function.
Right. The largest is usually the nucleus, the cell's control center. It houses the genetic information, the DNA, organized into chromatin. It's surrounded by a double membrane featuring nuclear pores that selectively control what gets in and out, things like RNA transcripts leaving for protein synthesis or specific proteins entering to regulate gene expression.
Surrounding the nucleus is the endoplasmic reticulum, or ER. It's this vast network of interconnected tubules and sacs. The rough ER is studded with ribosomes, that's where it gets its name, and it's the primary site for synthesizing proteins destined for membranes or secretion. The smooth ER, by contrast, is involved in lipid synthesis and acts as a major calcium reservoir within the cell.
Then there's the Golgi complex, which looks a bit like a stack of flattened sacks or pancakes. This organelle is like a sophisticated processing and sorting station for proteins. It ensures they mature correctly and are routed to their precise cellular destinations.
And what about the cell's power plant, the mitochondria? That's where the oxygen-dependent ATP production happens, right?
The mitochondrion is indeed remarkable. It has a double membrane, and the inner membrane forms these folds called cristae, which massively increase the surface area for energy production. Beyond generating ATP, it actually possesses its own circular DNA, a relic of its evolutionary past, and serves as a vital calcium reservoir. It also plays a central role in apoptosis, or programmed cell death, which is a crucial process for development and for eliminating damaged or potentially cancerous cells.
And for all the cell's internal waste management, it has the lysosome. This acts like the cell's recycling center, or maybe incinerator, filled with acidic enzymes to break down cellular waste, debris, and damaged organelles.
And the cytoplasm itself isn't just goo, it's highly organized by the cytoskeleton, a dynamic scaffolding of protein filaments. These act like internal beams, struts, and stays, providing structural support, determining cell shape, and enabling movement.
This scaffolding includes intermediate filaments. These are incredibly tough, providing structural support in areas of tensile stress, like where cells adhere to each other or to surfaces. The keratins in your nails and hair, those are examples of these resilient filaments.
Then you have microtubules, which are hollow polymers of a protein called tubulin. They are highly dynamic, constantly growing and shrinking, usually from a central point called a microtubule organizing center, or centrosome. They provide the framework for organelles like the ER and Golgi, are absolutely crucial for cell division, forming the spindle fibers that separate chromosomes and enable various forms of cellular motility.
How exactly do they drive motility?
Well, in structures like cilia and flagella, think of the tiny hairs that clear your airways, or the tail of a sperm cell. A molecular motor called dynein uses ATP energy to make microtubules slide past each other, causing bending. Within the cytoplasm, especially in neurons, another motor, kinesin, moves cargo like vesicles from the cell body out towards the axon tip, that's anterograde transport, while cytoplasmic dynein moves things back the other way, retrograde transport. This organized transport is vital, especially for cells with long extensions.
And finally, we have actin, or thin filaments, and myosin, which forms thick filaments. These are basically ubiquitous force-generating proteins. Actin filaments also grow dynamically, powered by ATP in a process called treadmilling.
Right. And myosin is a molecular motor that walks along these actin filaments, also using ATP. In muscle, myosin forms thick filaments that slide past thin actin filaments, causing muscle contraction. That's the classic example. But their roles extend far beyond muscle. They contribute to cell locomotion, form the core scaffolding of microvilli to increase surface area in epithelial cells, and create the contractile ring that pinches cells in two during cell division or cytokinesis.
So with all these proteins serving so many complex roles, how does the cell ensure they actually get to where they need to go, especially getting inserted into a membrane or secreted entirely without, getting stuck in the wrong place?
That's a really critical question, and the cell has an elegant solution called co-translational translocation. Proteins destined for membranes or secretion begin synthesis on ribosomes just floating in the cytoplasm. But a special signal sequence at the protein's beginning acts like an address label, or maybe a zip code. When this sequence emerges from the ribosome, a signal recognition particle, or SRP, binds to it, and that actually pauses protein synthesis temporarily.
Okay, pauses synthesis, then what?
Then this whole complex, the ribosome, the partially made protein, and the SRP docks with an SRP receptor on the surface of the rough membrane. This docking ensures the protein isn't fully made out in the cytoplasm if it's really meant to be embedded in a membrane or secreted. Once docked, protein synthesis resumes, but now the growing protein chain threads through a channel called a translocon, essentially a protein tunnel in the membrane. Inside the lumen, the signal sequence is usually cleaved off and the protein begins to fold.
But how does it stop if it's supposed to be a transmembrane protein sticking through the membrane?
Good question. There are other sequences within the protein hydrophobic stop transfer sequences. When one of these enters the translocon, it signals the channel to stop translocation and actually release that segment laterally into the lipid bilayer. That segment then becomes a stable transmembrane domain. By combining these start and stop signals, cells can build really complex, multi-spanning membrane proteins with specific orientations.
Okay, so once inside the proteins aren't quite finished. They undergo further maturation and, importantly, quality control, right?
Precisely. Proteins in the lumen undergo post-translational modifications. One common one is N-linked glycosylation, adding complex branched sugar chains. Another is disulfide bond formation, which helps stabilize their structure. And crucially, the is where molecular chaperones, which are helper proteins that use ATP, assist in proper protein folding, ensuring they achieve their correct three-dimensional or tertiary structure, and sometimes assemble into multi-subunit or quaternary complexes.
The cell's quality control here sounds pretty rigorous. Misfolded or improperly assembled proteins don't just get ignored.
Absolutely not. Misfolded proteins are typically retained in the They get tagged with a special molecular marker called ubiquitin, almost like a cellular return to sender, or maybe dispose of this label. Then they are retrotranslocated back out into the cytoplasm and fed into the proteasome, which is the cell's sophisticated protein degradation machine, kind of like a molecular shredder. This whole process is called associated degradation, or ERAD, and it ensures only correctly formed and functional proteins proceed further down the line.
From the these properly folded proteins embark on a highly organized journey known as the secretory pathway. I think George Palladino won a Nobel Prize for figuring this out using pancreatic cells.
That's right, a landmark discovery. This journey involves carrier vesicles budding off from specialized regions of the and fusing with the Golgi complex. Proteins then move sequentially through the different stacks, or cisternae, of the Golgi. As they move, they undergo further modifications, such as the intricate remodeling of those N-linked sugar chains we mentioned. It's like an assembly line, where different enzymes in each Golgi compartment add or trim specific sugars, creating unique glycoprotein patterns.
And proteins take different routes from the Golgi, depending on their ultimate purpose, correct?
Exactly. There's the regulated pathway for proteins that need to be stored in specialized secretory vesicles, think hormones or digestive enzymes, and they're only released by exocytosis when the cell receives a specific trigger signal. Then there's the constitutive pathway for proteins that are continuously delivered to the cell surface, perhaps to become part of the plasma membrane itself, or are immediately released from the cell without waiting for a signal. Carrier vesicles are essential for both pathways, budding off from one compartment and fusing with the next, all while carefully preserving the protein's membrane topology.
It's incredible how precisely these vesicles form and then find and fuse with the correct target membrane. How does that work?
It relies on an intricate network of specialized proteins. Coat proteins, like the famous clathrin, which forms this cage-like structure, help shape the membrane into vesicles, which then pinch off, often with help from another protein called dynein. For targeting and fusion, you have snares, V snares on the vesicle, and T snares on the target membrane, which recognize each other and effectively zip the membranes together. This whole process is tightly regulated by small Rab-GTP binding proteins acting like molecular switches. It's a really sophisticated choreography.
So beyond sending things out via exocytosis, cells are constantly bringing things in through endocytosis, which is essentially the reverse process where the plasma membrane invaginates to form vesicles. This is how cells import large nutrients, terminate hormone signals by internalizing receptors, recycle membrane components, and even clear pathogens.
Right. And we can distinguish different types. There's fluid phase endocytosis, which is a relatively nonspecific uptake of dissolved extracellular fluid and whatever happens to be in it, often using those clathrin-coated vesicles. But then there's receptor-mediated endocytosis, which is highly efficient and specific. Target molecules bind to specific receptors on the cell surface. These receptors then cluster together in clathrin-coated pits, and the whole complex is internalized.
And once inside, these materials go to the endosome. What happens there?
The endosome is a crucial sorting station. It has an acidic internal pH, which typically causes the ligands the molecules brought in to dissociate from their receptors. From there, the pathways diverge. Often, the ligands are sent on to the lysosome for degradation, while the receptors are packaged into different vesicles and recycled back to the plasma membrane, ready to bind more cargo. The LDL receptor, which brings cholesterol into cells, is a classic example of this efficient recycling system, driven by the pH difference between the outside and the endosome.
This precise cellular sorting has direct clinical relevance, doesn't it?
Yeah.
I know we see this in certain genetic conditions.
Absolutely. Disruptions in these pathways lead to lysosomal storage diseases. For instance, in eye cell disease, there's a mutation in an enzyme needed to add the correct address label, a mannose 6-phosphate tag, to lysosomal enzymes in the Golgi. As a result, these enzymes get secreted outside the cell instead of delivered to the lysosome. This leads to a buildup of undigested material inside lysosomes, causing widespread problems.
Wow. And Tay-Sachs.
Tay-Sachs disease is another tragic example, caused by a defect in a specific lysosomal enzyme responsible for breaking down certain lipids. These lipids accumulate particularly in neurons, leading to severe neurological damage. So understanding these intricate cellular sorting pathways is absolutely critical for diagnosing and hopefully one day developing effective treatments for these devastating conditions.
So we've explored all these basic cellular elements, membranes, proteins, organelles, transport systems. But what's truly astonishing is how these shared components combine and specialize to create the vast diversity of cell types in our bodies: neurons, muscle cells, epithelial cells, and so on. This process is called cell differentiation.
Exactly. And this specialization isn't really about inventing brand new molecular machinery for each cell type. Instead, it's about selectively expressing, organizing, and regulating a specific subset of molecules from the vast repertoire that all cells potentially possess, based on their shared genome. All this differentiation originates from pluripotent stem cells, which have the amazing capacity to give rise to many, if not all, of the distinct cell types in the body.
Let's focus on epithelial cells as a prime example of this specialization. They form sheets that act as dynamic barriers between your internal compartments and the external environment, or between different internal compartments. They allow for incredibly precise regulation of transport across these barriers.
Epithelial cells have several really unique and characteristic features that enable this function. First, they exhibit polarity. This means they have distinct functional domains. An apical membrane, which faces the lumen, or the outside world, often covered in microvilli to increase surface area for absorption or secretion, and a basolateral membrane, which faces the underlying tissue and the bloodstream. These two domains have different sets of proteins and lipids tailored to their specific roles.
And they're tightly connected to each other, forming a continuous sheet. Those connections are key, right?
Absolutely crucial. They have elaborate junctional complexes that link them together and maintain that polarity. These include tight junctions, which are found nearest the apical surface. They act like a seal or a fence, physically separating the apical and basolateral membrane domains and, very importantly, blocking the free diffusion of substances between the cells, the pericellular pathway. The tightness of these junctions can vary enormously depending on the epithelium. Some, like in parts of the kidney tubule, are relatively leaky, while others, like in the bladder, are virtually impenetrable.
What other kinds of junctions are there?
Just below the tight junctions, you typically find adhering junctions. These form a continuous belt around the cell where proteins called coherins link adjacent cells. These junctions are connected internally to the actin cytoskeleton, providing mechanical strength and potentially signaling cues. Then there are gap junctions, which are completely different. They form direct channels between the cytoplasm of adjacent cells, allowing small molecules and ions to pass freely. This electrically and metabolically couples the cells, allowing them to function as a coordinated unit. These junctions are often regulated, for instance, closing if a neighboring cell gets damaged.
And desmosomes.
Ah, yes, desmosomes. Think of these as incredibly strong spot welds or rivets. They occur at specific points, tightly holding cells together. Crucially, they link to the intermediate filament cytoskeleton inside each cell, creating a tissue-wide network that distributes mechanical stress. You find lots of desmosomes in tissues that experience significant physical forces, like skin or heart muscle.
This really highlights how the precise distribution of proteins, like we said, NAK pumps always on the base lateral side and absorptive epithelia, digestive enzymes always on the apical side in the gut, is absolutely essential for that directed vectorial transport across the epithelial sheet. It shows how all these cellular components we've discussed, the membrane, the proteins, the junctions, the cytoskeleton, work together in a meticulously coordinated fashion to enable complex and vital physiological functions throughout your body. So there you have it. Wow. From the basic structure of the plasma membrane, that incredible dynamic outer shield, to the diverse roles of its proteins acting as gates, signals, and anchors. Then moving inside to the specialized functions of organelles and the dynamic cytoskeleton providing structure and movement. And finally, seeing how these components are meticulously synthesized, sorted, delivered, and come together in specialized cells like epithelia to perform vital functions for the whole organism. It's an incredibly intricate dance.
And that's exactly what it is. Understanding these foundational cellular mechanisms truly is the big picture in physiology, wouldn't you say? It reveals how these microscopic processes underpin every single macroscopic body function, from the way your nerves fire and your muscles contract, to how you digest food, and critically, how disruptions in these very processes contribute to disease. It all starts at the cell level.
You've just taken a deep dive into some truly complex physiological concepts, and hopefully you followed along. You are absolutely part of the last minute lecture family, and you're definitely capable of mastering this material.
Yeah, absolutely. And maybe leave you with this thought. Consider the intricate dynamic dance of molecules and structures within just a single cell, the stuff we've just uncovered. Think about how all that allows it to respond and adapt constantly to the ever-changing, sometimes quite hostile environment that is life itself. It makes you wonder, what new adaptations might cells evolve to face future environmental pressures?
That's a profound thought to ponder. Keep diving deeper into physiological knowledge with us. We'll see you on the next deep dive.